In this session, we are going to talk about pK, the negative log of equilibrium constant of a chemical reaction. In the first part of my session, we will discuss about the reversibility of a chemical reaction, the concept and nature of chemical equilibrium, and the mathematical expression and significance of equilibrium constant K. In the second part of my talk, I will be focusing on the biological aspects of chemical equilibrium with special reference to biologically important acids and macromolecules. In a simplified chemical reaction where A and B reversibly combines to form C and D, the rate of forward reaction Vf is expressed as Kf times concentration of A times concentration of B, where Kf is the velocity constant of forward reaction. Similarly, Vr, the rate of reverse reaction, can be expressed as Kr times concentration of C times concentration of D. Now, equilibrium is defined as a condition where the velocity of forward and reverse reaction equals each other. Or in other words, at equilibrium, Vf equal to Vr, or Kf times concentration of A times concentration of B equals Kr times concentration of C times concentration of D. Equilibrium constant K is defined as the ratio of the rates of forward and reverse reaction, or in other words, K equal to Kf by Kr, which in turn equals to concentration of C times concentration of D divided by concentration of A times concentration of B. In a more generalized reaction, if A moles of substance A reacts with B moles of substance B to form reversibly C moles of substance C and D moles of substance D, then the equilibrium constant K is defined as C to the power of C times D to the power of D divided by A to the power of A times B to the power of B. Physically, the condition of equilibrium may be depicted by the analogy of a vessel attached to two water taps. One delivers water into the vessel while the other drains out. If the flow rates of the two taps are equal, then at any given point of time, the net amount of water delivered into the vessel equals the amount of water delivered out of the vessel, and the net quantity of water in the vessel remains unchanged. The figure shows the change in rates of forward and reverse reaction with time. At the beginning of the reaction, the rate of forward reaction is high and that of the reverse reaction is slow. This is simply because of the fact that at the beginning of the reaction, the concentration of the reactants are high while that of the products are low. But with the passage of time, the concentration of the products build up and the rate of reverse reaction goes high and the forward reaction goes down. At equilibrium, the two rates become equal. However, at equilibrium, the relative concentration of the reactants and the products need not necessarily be the same. This depends on the value of K, the equilibrium constant. If the value of K is significantly greater than 1, then the amount of product is much more than the amount of reactants. While if the value of K is significantly less than 1, then the reactants accumulate in the mixture while the product concentration stays low. However, at equilibrium, the relative concentration of the reactant and the product need not necessarily be the same. This depends on the value of K. If the value of K is significantly greater than 1, then formation of product is favored, and in the reaction mixture, the amount of product is much more than the amount of reactants. If the value of K is significantly less than 1, then the concentration of reactants in the mixture is much higher than that of the product. There are factors that affect the equilibrium constant K. Temperature. In the case of an endothermic reaction, the value of K increases with increase of applied temperature, and the opposite effect is observed for an exothermic reaction. The pressure dependence of equilibrium constant is usually weak in the range of the pressure normally we work in. However, if the reaction results in change of number of moles, then pressure can have a significant effect. The value of K spans over many orders of magnitude. Hence, a logarithmic scale is in practice. The logarithmic constant pK, which is equal to minus log K to the base 10, is called the negative log of equilibrium constant. The understanding of equilibrium constant is at the heart of many biological processes. We frequently use words like stability constant, formation constant, binding constant, association constant, dissociation constant. These are all equilibrium constants in different names. In the second part of my discussion, I will focus on the dissociation constants of biologically important acids. An acid dissociation constant Ka, also known as the acidity constant or acid ionization constant, is a quantitative measure of the strength of the acid in solution. It is the equilibrium constant of the dissociation reaction, and for a generalized acid Ha, it can be written that Ha reversibly dissociates to form A minus, the conjugate base, and the proton H plus. Now Ka equals concentration of A times concentration of H divided by the concentration of undissociated Ha, and pKa equals minus log Ka to the base 10. There are several methods for the determination of pKa. For example, the Poisson-Boltzmann equation, the empirical methods, and molecular dynamics based methods. However, pKa can be measured easily in laboratory by potentiometric titration method. Amino acids are biologically important acids. They are the building blocks of proteins. Amino acids are so named because they bear an amino group and a carboxylic acid group on the same molecule. Both these amino groups and the carboxylic acid groups can donate or accept protons. However, that depends on the context. Now, when and under what condition these groups will donate or accept protons? To know this answer, we have to go to the titration curve. As can be seen from this graph, at lower pH, the glycine molecule exists as the fully protonated form, that is, its amino group exists as NH3 plus, while its carboxylic acid group exists as COOH. As the pH rises, the carboxylic acid group starts to donate its proton first. The donation of protons starts around pH 1, and by pH 3, the donation of proton by the carboxylic acid group is complete. And the midpoint of transition of this donation of proton by the carboxylic acid group is at 2.34 pH, or in other words, the pK1 is 2.34 for glycine. The pK2, that is, the pH at which the NH3 plus group of glycine starts to donate its proton, starts around pH 8 to around pH 11, and the midpoint of transition lies at around pH 9.6, or in other words, the pK2 of glycine, that is, the pH at which the NH3 plus group of glycine donates its proton, is pH 9.6. Isoelectric point Pi is the pH at which a particular molecule or a surface is electrically neutral. That means in an amino acid, both the carboxylic acid group and the amino group can donate protons. Pi is calculated as pK1 plus pK2 by 2. The table here shows the pK1 and pK2 value of all the 20 amino acids that naturally occur in proteins. The amino acids which are monocarboxylic and monoamino, that is, they contain only one amino group and one carboxylic acid group. The Pi is calculated as the mean of pK1 and pK2. However, there are amino acids which contain carboxylic acid group or NH2 group at the side chain. For example, glutamic acid has an extra COOH group other than the alpha carboxylic acid group at its side chain. The Pi of glutamic acid is calculated as the mean of the pK value of alpha carboxylic acid group and side chain carboxylic acid group. Similarly, lysine and arginine are amino acids that carry an extra NH2 group at their side chains. Pi of lysine and arginine are calculated as mean of the pK value of amino groups present at the alpha position and at the side chains. Monoprotic acids are acids that produce one proton per molecule of acid. pKa equals minus log Ka to the base 10. If we replace the value of Ka as concentration of A times concentration of H divided by concentration of Ha, and pH equal to minus log H to the base 10, we get pH equals pK plus log concentration of A divided by concentration of Ha. This is called the Henderson-Hasselbalch equation. Since pH equal to pK plus log concentration of A minus divided by concentration of Ha, when the acid is dissociated halfway, A minus equals Ha, then pH becomes pKa. Or in other words, pKa, or the acid dissociation constant, is the pH at which the acid is dissociated halfway. If we look at the curve which depicts the percent formation of HA and A minus with respect to the difference of pH and pK, we see that when pH minus pK is minus 2, the concentration of HA is higher than that of concentration of A minus. When pH minus pK is 0, that means pH equal to pK, we see the population is half and half, that is 50% is occupied by HA and 50% by A minus. The situation is interesting in polyprotic acids, the acids that donate more than 1 protons per molecule of acid. Phosphoric acid present in DNA is one such good example. Phosphoric acid can donate 3 protons and the respective pK values are 2.15, 7.20 and 12.37. The graph that shows percent formation of different species with respect to pH, we see that at lower pH the form H3PO4 predominates. Upon increase of pH, H2PO4 minus increases and H3PO4 decreases. When the pH increases further, there is concomitant formation of HPO4 2 minus and PO4 minus 3. Normally, for a polyprotic acid, the value of pK1 is less than that of pK2, which is further less than that of pK3. It's interesting to note that for phosphoric acid, at pH 3, there is 50-50 population of H3PO4 and H2PO4 minus. However, at pH 6, there is only a single population of H2PO4 minus. And at pH 10, there is a single population of HPO4 2 minus. There are several biological importances of knowing pKa value. The concept of pK is applicable in many different areas of chemistry, biology and medicine. Many medicines are actually weak acids or weak bases, and a knowledge of their pK value can be used to estimate the extent to which they are absorbed in blood and body fluid. Acid-base homeostasis and kinetics of many reactions are dependent on the pK value of acids and bases used. pK values of amino acids help in understanding the folding process and function of proteins and other macromolecules. There are several softwares for determination of protein pKa values. Accelera's pKa, H++, MCCE, Carlsberg Plus, PKD server and PropKa. These are some of the softwares that are used by different laboratories to calculate pK values of proteins. We come to the concluding section of this talk, and the take-home messages are: most of the biochemical reactions are reversible in nature. Reversible chemical reactions attain equilibrium. Equilibrium constant K is defined as the ratio of the rate constants between forward and reverse reaction. At equilibrium, the velocity of the forward and the reverse reaction are equal. However, at equilibrium, the relative concentration of the reactants and products may vary depending on the value of K. If K is significantly greater than 1, then product accumulates while when K is significantly less than 1, reactants accumulate and the amount of product is low. Since the value of K spans over several orders of magnitude, in practice a logarithmic scale is used. pK equals minus log K to the base 10. The knowledge of equilibrium constant and pK is instrumental to the understanding of several biological processes. pKa, or the acid dissociation constant, indicates the quantitative behavior of an acid in solution. Knowledge of pKa helps in understanding the structure and function of biological macromolecules like DNA and protein.